Okay, you're fine. All right,
Hi, my
Genevieve.
name is Genevieve Johnson. I'm a sophomore here at Westside High School. Today is December 30th, and my project is an assay of beta-galactosidase. The question I wanted to ask is, can beta-galactosidase activity be measured with a spectrophotometer? Now, I know some of these words aren't familiar with you. By the end of my project, you should know what each of them mean. My hypothesis was that beta-galactosidase activity could be measured with a spectrophotometer. I got this hypothesis from Dr. Claiborne Glover. He's a professor at UGA in molecular biology, and I've got this from his discussions I had with him. Two purposes were in my project. The first purpose was to determine whether beta-galactosidase activity could be measured with a spectrophotometer, as well as demonstrate how to perform beta-galactosidase assays as a prerequisite for doing research into gene expression. There's a picture of the spectrophotometer screen and a DNA screen, or two different screens. My project reports beta-galactosidase activity done during enzyme assays, where beta-galactosidase is the enzyme and MPG is the substrate. The first picture is a picture of beta-galactosidase, the enzyme, and over here is a picture of lactose. Now I have a picture of lactose up here because in my project I'm using MPG as a substitution for lactose, and I'll get into that in a little bit. Here's some terms and concepts I thought would help, the answers would help y'all understand better. What is an enzyme? What is beta-galactosidase? What are lactose, galactose, and glucose? What is beta-galactosidase activity? What is a spectrophotometer, and what is MPG? And last, how is doing beta-galactosidase enzyme assay a prerequisite for research into gene expression? What is an enzyme? All enzymes are protein, and they decrease the time it takes for a reaction to occur. Reactions occur with a lower activation energy. Here's an analogy. The first picture is an uncatalyzed reaction without an enzyme, and the next picture is a catalyzed reaction with the enzyme. The brown barrier represents a high activation energy in the first picture and a low concentration in the second picture. The waves have a hard time going over the barrier because the activation energy is high. But in the next picture, the waves are able to go over the barrier because the reaction is low, and because the activation energy is low, and the reaction can happen quicker. Since enzymes are proteins, they are long chains of amino acids. There's 20 types of amino acids, and the only difference between each amino acid is the side chain. And there's less of them. I don't want to read them. Enzymes bind substrates in their active site. A reaction occurs in the active site. They then catalyze the reaction and release the product of the reaction. In this picture, the red is a substrate and the blue is an enzyme. The substrate binds with the enzyme in an enzyme-substrate complex. It then becomes an enzyme product complex when the substrate is cleaved. Now, cleave means cut. The end result is the enzyme and the product, too. Beta-galactosidase has 4 domains. Each of the colors in this picture represents a different domain. Within each of these domains is 1,023 amino acids, and each domain has an active site. Without each of these domains, the reaction could not happen. The enzyme would not work because the enzyme would either change shape or wouldn't have all 4 active sites to make a reaction. Beta-galactosidase catalyzes the reaction of lactose plus water to produce galactose and glucose. There's beta-galactosidase and the reaction happens. What is beta-galactosidase? To help understand this more, what are lactose, galactose, and glucose? To answer that question, I'll answer the simpler question of what is H2O. Now, what is H2O? When you ask this, you'd probably say water. And you'd be correct. Another question. What is C6H12O6? Look at this question. You'd probably say glucose. You might even say galactose. You'd be right. You could even say mannose, and you'd be right too. These three molecules have the same chemical formula. And this is called an isomer. But what is the difference between each of these isomers if the chemical reaction is the same? The chemical formula is the same. The answer is spatial arrangement. The spatial arrangement of the hydrogen atom and the hydroxyl group on the number 2 and number 4 on the number 2 and number 4 carbon atom. Now, a hydroxyl group is hydrogen and oxygen together. A hydroxyl group works either OH or HO, and it works going up and down. Mannose is not relevant to my project, but it is the other isomer to these two. As you can see, mannose on the number 1 carbon, which is green, has the hydroxyl group different from each of the others, and on the number 4 carbon in galactose is different from the others. Now, what are lactose, galactose, and glucose? Here are the two, galactose and glucose. Each of these are 6-carbon sugars. This larger picture over here shows these 2 blue 6-carbon sugars at the top. Each 6-carbon sugar is a monosaccharide, and a monosaccharide is 1 molecule. These 2 monosaccharides together, when one molecule is taken away from these, the two bond together in a glycolysic bond. This glycolysic bond makes these 2 molecules become disaccharide, 2 molecules. The disaccharide is now lactose. Here's the reaction. Lactose plus water yields galactose plus glucose. And here's an illustrated picture of that. To give the full effect of what beta-galactosidase is, I'm going to tell you where galactose comes from and where beta comes from. But to get that, you have to know how molecule names and enzyme names are formed. When a molecule contains a galactose molecule binded with a second molecule, that is called a galactoside. Enzyme names are formed by adding the suffix ASC to the end of the substrate name. That the enzyme catalyzes. Lactose is therefore a galactoside, and the enzyme that it catalyzes is therefore a galactosidase. Here's a picture of the bond. Now, what does the beta in beta-galactosidase mean? When the galactose molecule and the galactose molecule and the second molecule bind together, and when the hydroxyl group is in the beta position, the resulting molecule is a beta-galactoside. Now, the hydroxyl group, as I said, is hydrogen and oxygen. And the way it works is just going up and down. Up being in a beta position and down being in an alpha position. The hydroxyl group freezes in that beta position and results with the beta-galactosidase. And it links. There's a picture of it.